Welcome to Pharmacology Insights, where we break down complex pharmacology into simple, easy to understand concepts. 1.4 billion. That's how many people worldwide are living with high blood pressure right now. This is not just statistic tucked in a footnote. It's the single largest modifiable risk factor for strokes, heart failure, kidney disease, and premature death. And here's the frustrating part. We already have good drugs. ACE inhibitors, angiotensin receptor blockers, calcium channel blockers, diuretics, and beta blockers. Decades of pharmacology, billions of prescriptions. So, why are so many patients still failing? In the United States alone, roughly half of all patients taking multiple blood pressure medications still have persistently elevated readings. And within that group sits a harder subset. Patients classified as having resistant hypertensives, whose blood pressure remains uncontrolled on 3 or more drugs, including a diuretic. So, what's going on? Why is the standard toolkit failing these patients? The answer, in large part, comes down to one hormone: aldosterone. If you're a student or clinician, you know the renin-angiotensin-aldosterone system, the RAAS. But let's walk through it quickly, because this is the pathway where Baxdrostat strikes. When blood pressure drops or sodium runs low, the kidneys release an enzyme called renin. Renin cleaves a circulating protein called angiotensinogen into angiotensin 1. Angiotensin 1 then gets converted by angiotensin-converting enzyme, or ACE, into angiotensin 2, the main effector hormone of the system. Angiotensin 2 does a few things. It constricts blood vessels directly. And it signals the adrenal glands, the two small structures sitting on top of your kidneys, to produce aldosterone. Aldosterone is a mineralocorticoid hormone. Its job is to act on receptors in the kidney and trigger the epithelial sodium channel to pull sodium back into the bloodstream. Water follows sodium and blood volume rises. Higher blood volume means higher blood pressure. In a healthy person, this is a tightly regulated feedback loop. But in many patients with resistant hypertension, aldosterone levels are chronically and inappropriately elevated. Sometimes from primary aldosteronism, sometimes from dysregulated RAAS activity. And that excess aldosterone is quietly keeping blood pressure elevated, no matter how many drugs gets added to the regimen. So have we tried to fix this? Yes, in quite a few ways. ACE inhibitors and angiotensin receptor blockers work earlier in the RAAS cascade, reducing angiotensin 2. And with it, the signal to produce aldosterone. They work well, but they don't completely shut down aldosterone. The body has backup pathways, and aldosterone can still climb. Mineralocorticoid receptor antagonists, or MRAs, takes a different approach. Drugs like spironolactone and eplerenone don't reduce aldosterone itself. They block the receptor it binds to. No receptor activation, no sodium reabsorption. Spironolactone is actually guideline recommended as a fourth-line agent in resistant hypertension, and for good reason. It works. But here's where the story gets complicated. Spironolactone is a non-selective steroid compound. It doesn't just block mineralocorticoid receptors. It also cross-reacts with androgen and progesterone receptors, triggering gynecomastia in men, breast tenderness, menstrual irregularities, and sexual dysfunction. These aren't minor inconveniences. They're the main reasons patients stop taking the drug. And layered on top of that is hyperkalemia, dangerously high potassium, a real concern with any RAAS acting agent. Eplerenone is more selective. but still has efficacy limitations. The dream has always been, what if instead of blocking the receptor, we just stopped aldosterone from being made? Here's where the pharmacology gets really fascinating, and where a challenge that held this field back for decades becomes clear. Aldosterone is synthesized in the zona glomerulosa of the adrenal cortex. The final step in the process, the conversion of 11-deoxycorticosterone to aldosterone, is catalyzed by an enzyme called aldosterone synthase, encoded by the gene CYP11B2. So the logic is: inhibit aldosterone synthase, block aldosterone production, lower blood pressure. Simple, right? Except there is a pharmacological booby trap in the adrenal cortex. Right alongside aldosterone synthase sits a closely related enzyme, 11-beta-hydroxylase, encoded by CYP11B1. This enzyme is responsible for the final step of cortisol synthesis. Aldosterone synthase and 11-beta-hydroxylase share 93% sequence similarity. They are, structurally, nearly identical. When early aldosterone synthase inhibitors were developed, they simply couldn't tell the two enzymes apart. Block one, block the other, which meant suppressing cortisol along with aldosterone. Cortisol is not a hormone you want to mess with. Deplete it, and you risk adrenal insufficiency. The first clinical aldosterone synthase inhibitor candidate, Aldosterone, ran into exactly this problem. It was ultimately repurposed to treat Cushing's syndrome, a condition of excess cortisol, rather than hypertension. For years, that 93% similarity problem was considered an almost insurmountable barrier to developing a safe aldosterone synthase inhibitor for hypertension. In May 2026, the FDA approved Baxdrostat, a first-in-class, highly selective, potent, oral aldosterone synthase inhibitor. Originally developed by Roche and later licensed to Syncor Pharma. Now part of AstraZeneca, Baxdrostat is a small molecule, taken orally, that is engineered with exquisite selectivity for aldosterone synthase over 11-beta-hydroxylase. How does it achieve this selectivity despite 93% structural overlap? The answer lies in subtle differences in the binding pocket geometry between the two enzymes. Baxdrostat was specifically designed, through iterative medicinal chemistry, to exploit those small structural differences and bind with high affinity to aldosterone synthase, while having significantly reduced interaction with 11-beta-hydroxylase. The downstream effects are clean and logical. Baxdrostat selectively inhibits aldosterone synthase in the adrenal zona glomerulosa. Aldosterone synthase activity falls, and aldosterone production drops in a dose-dependent manner. With less circulating aldosterone, there is less stimulation of mineralocorticoid receptors in the kidney. Less epithelial sodium channel activation means less sodium reabsorption, less water retention, and reduced blood volume. Blood pressure falls. And critically, cortisol levels are preserved. Preclinical studies in cynomolgus monkeys confirmed that Baxdrostat inhibited aldosterone synthesis, without affecting ACTH-stimulated cortisol rises. This was replicated in healthy human volunteers in phase 1. Now, let's look at the trial that convinced the FDA. The phase 3 BaxHTN trial enrolled 796 patients with uncontrolled or resistant hypertension, who were already taking at least 2 other antihypertensive agents. That's the key. These weren't untreated patients. They were already medicated and still not controlled. Participants were randomized 1:1:1 to receive Baxdrostat 2 mg, 1 mg, or placebo once daily, added on top of their existing regimen. The primary efficacy endpoint was the change in baseline and seated systolic blood pressure, or SBP, at week 12. At 12 weeks, systolic blood pressure dropped a mean of 14.5 mm of mercury with the 1 mg dose. and 15.7 millimeters of mercury with a 2 mg dose, compared with 5.8 millimeters of mercury in the placebo group. That translates to placebo-adjusted reductions of 8.7 millimeters of mercury and 9.8 millimeters of mercury, respectively. Both statistically significant at p less than 0.001. A placebo-adjusted systolic reduction approaching 10 millimeters of mercury is not a modest signal. This is the range where we expect to see real reductions in cardiovascular events. Results were consistent across both uncontrolled and treatment-resistant subgroups. Whether patients were on 2 drugs or 3, Baxdrostat worked. And to put a human face on those numbers, roughly 40% of patients in the Baxdrostat groups achieved blood pressure control, with systolic blood pressure reading under 130 millimeters of mercury, compared with 18.7% in the placebo group. For a population that had already failed multiple medications, that's a remarkable result. Baxdrostat was generally well tolerated, with no unexpected safety signals. The adverse effects observed were consistent with its mechanism of action, which is exactly what you'd predict when you reduce aldosterone's influence on the kidney. Hyperkalemia, or elevated potassium, occurs in up to 10.2% in the 2 mg group across pooled analyses. This is logical. Less aldosterone means the kidneys excrete less. Monitoring is important, particularly in patients already on other RAAS-blocking drugs. Other effects included hyponatremia, hypotension, muscle spasms, and dizziness. No meaningful impact on kidney function or cortisol levels was observed, validating the selectivity profile established in earlier studies. The FDA approved Baxdrostat under its brand name Baxfendy, marks a genuine first. No aldosterone synthase inhibitor had ever reached approval for hypertension before this, anywhere in the world. The approved indication is add-on treatment for adults with hypertension not adequately controlled on existing medications. Precisely the population studied in BaxHTN, and precisely the population that has had the fewest good options. So how should we think about Baxdrostat's place in clinical practice? For now, it is an add-on therapy, a tool to reach for when standard regimens haven't brought blood pressure to goal, particularly where elevated aldosterone is suspected as the driver. It isn't replacing ACE inhibitors or calcium channel blockers. It's complementing them, filling a gap that has been open for a long time. One nuance worth noting is that head-to-head data comparing Baxdrostat directly against spironolactone are not yet available. That comparison will be important. But for patients who can't tolerate spironolactone's hormonal side effects, Baxdrostat already offers a compelling alternative. A mechanism that achieves similar blood pressure benefit, without the androgen and progesterone receptor baggage. Additional phase 3 trials are underway, and the pipeline is expanding. Step back for a moment. The last genuinely novel mechanism to enter mainstream hypertension therapy was the direct renin inhibitor Aldosterone, approved in 2007. Before that, angiotensin receptor blockers in the late 1990s. For nearly 2 decades, we've been writing the same pharmacological prescriptions. Different combinations, same mechanisms. Baxdrostat is not a refinement of something that already exists. It's a first-in-class drug that finally solves a puzzle that eluded pharmacologists for more than 30 years. The 93% structural overlap between aldosterone synthase and 11-beta-hydroxylase wasn't just a scientific inconvenience. It was a drug discovery wall that required a fundamentally new approach to molecular design to get over. For students, this is a masterclass in target selectivity. Knowing your enzyme's binding pocket at atomic resolution isn't an academic exercise. It's the difference between a drug that reaches patients and one that gets abandoned or repurposed. For clinicians, this is a new mechanism for a population that has been running out of options. For patients who've been told their blood pressure is treatment resistant, this is real, evidence-based hope. And it has been a long time coming.